Must be starting chapter 13, but we're still in chapter 14. All right, are there any questions before we begin? Can somebody confirm that you can hear me?
Yes.
Great. Come on. How we go? All right, let me share my screen. Are there any questions?
I did have a quick question about the quiz last night.
Uh-huh.
Um, when it was due by 11:59, right?
Uh-huh.
Okay, because I took it about 7:45, and at 8 o'clock, a thing popped up during the quiz saying that it was going to be submitted late. So it says next to my submission and my score that says late.
Don't worry about it as long as you got it in. I'm not sure what happened, but I'll take a look at it
Okay.
after the lecture.
Okay, thanks.
All right. Are you seeing my screen?
Yes.
All right, great. So today we're supposed to be starting chapter 13. We're a little behind schedule. I need to finish up chapter 14. There is no lecture today, but do note that the unknown notes pages are going to close on Friday, December 1st, and the unknown project is due Saturday, December 2nd. The last chance to ask will be Friday, but you must have a test that takes a day if you put in a request on Friday. All right, let's get going. Now I've got that up. Where is that? Here it is. So vector transmission we mentioned was when an animal carried the disease to a human. Let's talk a little bit about nosocomial infections. Nosocomial infections are one of the most important topics that you can learn in microbiology, assuming that you're going to go into the healthcare industry, becoming a nurse or something working in a hospital-like setting. Nosocomial infections are also called hospital-acquired infections. They are infections that a patient does not have until they stay in a hospital or a hospital-like facility, meaning the patient did not have it and then went into the facility and picked it up in the facility. About 5 to 15% of all hospital patients come down with a nosocomial infection. That's 2 million people a year in the United States. That's 20,000 deaths occurring due to a nosocomial infection in the United States per year alone. It used to be the 8th leading cause of death in the United States. I don't know if that's still the case because of COVID-19. It may be the 9th leading cause now. This slide is showing you that there's a lot of cost to nosocomial infections. You don't need to know the exact numbers, but about 1 out of 10 hospital patients gets a nosocomial infection, according to the Centers for Disease Control. About 50% of these nosocomial infections are preventable. Nosocomial infections result from an interaction of 3 different factors. One, there are microorganisms present in the hospital environment. Hospitals are major reservoirs of patients who are sick, and sick patients frequently have infections that make the reason for why they're in the hospital. However, there are also nosocomial infections occurring by normal microbiota that are opportunistic pathogens giving the nosocomial infection. And these are also present in the hospital, in all of the patients, in all of the staff and the visitors. And then to make matters worse, in the hospital environment, there are some nosocomial pathogens which are antibiotic resistant. and it's a little more common to find an antibiotic resistant pathogen in a hospital because of the frequent use of antibiotics in the hospital. Any question about any of that? So there are microorganisms present in the hospital environment, and they can give a patient a nosocomial infection. Second, the patient coming into the hospital-like setting is often a compromised host, one whose resistance to infection is impaired either by disease, therapy, or by burns or trauma, meaning the patients may have broken skin or broken mucous membranes that will compromise the host. Burns, injections, ventilators, urinary catheters, the administration of anesthesia all make the patient more compromised to getting a nosocomial infection. Patients in the hospital-like setting can also be suppressed in their immune system, and that suppression can be by drugs, radiation therapy, burns, which can affect the immune system. All of these can weaken the immune system. It's well known that cancer drugs frequently weaken the immune system. There's also a chain of transmission in the hospital. The principal routes of transmission are direct contact transmission, such as patient to patient or doctor to patient. In special care units, hospital staff are trained to prevent the spread of nosocomial infections from patient to patient. But there's also indirect contact transmission via a fomite, such as on a urinary or IV catheter, or contaminated surgical dressings. And then in the hospital, there can be vehicle or airborne transmission, where the ventilation system can spread the nosocomial infection. I don't know if you remember, but gosh, it might be several years back now, there were a few patients who came down with Ebola, and they were treating Ebola in Africa, and they came to the United States because their tour of duty ended or they became sick with Ebola. And so they went into the hospital, and there was real concern that Ebola could be spread by the ventilation system. And so they took steps to prevent that in the American hospital. I think what they did was they either put special filters on the air collected from the room of the patients, because I'm sure they did not filter the entire air of all the air in the hospital, but the air being brought in to the ventilation system in the room, I think they put a filter on it to prevent the spread of Ebola in the United States from the patients who had it. Now, Ebola was mostly spread by direct contact transmission, but there was concern about it spreading by vehicle transmission. Any questions about the three reasons for why there's nosocomial infections in the hospital-like setting in the United States? There are different ways you can control nosocomial infections. And if you work in the hospital-like setting, you should learn these ways. There are certain procedures that are generally implemented. You use aseptic technique. You learn that in microbiology now. Now, when you go out into the hospital-like setting in your job, you practice those techniques always. You proper handle contaminated materials, so the contaminated material doesn't spread beyond the material that you're handling. You have frequent and thorough hand washing. In the hospital, we have the education of staff, and that began, I think, as early as the 1960s, at least among the doctors, maybe 1970s. It occurred to the other hospital staff. And then we began educating the patients and the visitors. It was a time when you went into the hospital, you guys probably aren't even old enough. There weren't signs up and there weren't sanitation devices inside the elevator or inside outside the patient's room saying, sanitize your hands to prevent the spread of disease. And like I said, in all hospitals now, there's signs and sanitation stations outside of every room and usually inside the elevators too. There are many other things you can do to prevent the spread of nosocomial infections, and you can see the online chapter notes for getting some of those other things. But like I said, now in the United States, the staff, the patients, and the visitors are all educated to prevent the spread of disease. And it's actually worked. The cases of nosocomial infection in the United States have now dropped by about 50% since, gosh, about 7 years ago. Any question about any of that? All right. So nosocomial infection is one of the worst things that can happen to a patient coming into the hospital. And then if it's a nosocomial infection, which is antibiotic resistant, the doctors don't know that at first, and they just give the standard antibiotic therapy, and then it's not responding. And so they switch to a different therapy. Well, guess what? The patient may be so sick that they don't have time. And that's why sometimes nosocomial infections kill patients. All right, let's move on to epidemiology, the study of where and when diseases occur and how diseases are transmitted in population. The CDC collects and analyzes U.S. epidemiological information. Some diseases are notifiable diseases, those that physicians are required by law to report to the health authorities. And this will be collected by the CDC and published weekly in the Morbidity and Mortality Weekly Report, usually abbreviated MMWR, which you can find at www.cdc.gov. Usually, a doctor or a nurse, when they see a patient with a notifiable disease, they report it to the local health facilities, which in the United States would be the county health facilities. And then the county passes it to the state. I'm not sure where the state offices are in Washington, but in Oregon, the state health facility is in Salem. I used to live in Oregon. And I said, I just don't know where it is in Washington. And then the state health offices, forward it to the Center for Disease Control. Any question about any of that? All right, two terms of epidemiology you should know. Morbidity is the incidence of a specific notifiable disease. The morbidity rate is the number of people affected in relation to the total population in a given time period. The mortality is the deaths from a specific notifiable disease. The mortality rate is the number of deaths from a disease in relation to the population in a given time period. Any question about any of those two terms? Any question about chapter 14 at all?
Nope.
Okay, let's go on then to chapter 13. Went too far. Chapter 13 is studying viruses, viroids, and what's the third one? Prions. The major goals are shown here. Know the terms of this chapter. 2, understand the characteristics of viruses, the structure of viruses, and the classification of viruses. 3, understand about viral multiplication. And 4, lastly, understand about prions and viroids. Any question about the overview and the brief outline? And the major goals, I guess, too. All right. The only goal a virus has is to replicate. How do they do that? A virus follows a general strategy. They invade their host cell, they hijack the cell, and then force the cell to make viral copies. The viral copies then escape, and then the virus cycle continues. And let's watch a little video, if I can get that to work, on how a virus replicates and takes over a cell. Saying what happened to the picture?
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
Okay, any questions about that video? Dump that. I'll keep it up in case that's the next video I'm going to show. Oop, what happened? Here are some general characteristics of viruses. Viruses are inert when they are not within a host cell. They display little or no metabolic activity outside of their host cell. Viruses require a living host cell to multiply. So you can say that viruses are obligate intracellular parasites, meaning they require a host cell to replicate in. And actually, outside of their host cell, they have no or little metabolic activity. But they do have metabolic activity inside their host cell. They multiply within their living host cell by using the host cell's enzymes, nucleic acid, amino acids, ATP, and etc., meaning they use the host cell machinery to replicate. Viruses have no ATP generating mechanism. They rely on the ATP from their host cell. Viruses contain either DNA or RNA, but never do they have both. The DNA or the RNA is their genetic material, which is their genome. And you'll note that viruses can have RNA as their hereditary material. That's unheard of in a host cell, in a regular cell, in a cell at all, for all that matter. Viruses have the genome as their central molecule, and surrounding that genome is a protein coat. Some viruses have around the protein coat a envelope, which is a lipid envelope, which would surround the protein coat. This virus has this side of the virus cut out so you can see inside the virus. Any question about viruses? The host range of a virus is the species that the virus can infect and replicate in. The host range is determined by the host attachment sites, such as a cell wall, flagella, etc., and cellular factors necessary for the viral multiplication. Viruses have specific molecules that bind to specific receptors on host cells. The receptors are just a molecule on the host cell that the virus binds to. And so that's the name of the molecule the virus binds to. The name of the molecule on the virus that binds to the host receptor is called either a ligand or an adhesion molecule. Most viruses only infect the cells of one or a few species, such as HIV only infects the cells, actually only infects certain cells of humans, and it does infect certain cells of one of the chimps. So it actually does infect cells of two different species, but it does not infect, say, like muscle cells or skin cells. It does infect only a few cells like macrophages and T helper cells. It may infect brain cells, I don't remember. There are some exceptions where there are viruses which infect cells of many different species. I think bird flu might be an example because that virus can infect many different birds, if not all birds, and it can infect at least some mammals, including humans. And so, like I said, there are some exceptions, but most viruses are very specific and they're very narrow in the cells that they can infect. Like HIV only infects a few cells of two different species. Any question about any of that? And that specificity is due to the adhesions or ligands on the virus that bind to the receptors on the host cell, the specific molecules of the virus that bind to the specific molecules of the host cell. Viruses in the relative size is shown here. This would be a fairly small eukaryotic cell. That's a red blood cell, and it's so large we're only seeing a little portion of it. That would be E. coli, which is a little larger than most bacteria. And there is the largest virus, lengthwise. There's the largest virus diameter-wise, megavirus. Ebola virus is actually the longest virus. It's not the biggest virus. Megavirus is the most commonly stated biggest virus by diameter. And then there's some which are quite a bit smaller. Let me blow this up. So smaller, smaller, smaller. Poliovirus is probably the smallest virus you've ever heard of, around 30 nanometers. There are some smaller viruses, but I'm sure you've never heard of them, like bacteriophage MS2, a little smaller than poliovirus at 24 nanometers. So when we're talking about viruses, it's important to talk about their viral structure. As I mentioned, viruses always have a nucleic acid for their genome, either RNA or DNA. It's a central molecule. Viruses are always encapsulated by a protein coat. And that makes some viruses are just that way. And then there are other viruses that are surrounded by a lipid envelope. A virion is a single mature, complete infectious virus particle. The nucleic acid of a virus can be either DNA. If it's DNA, it can be double-stranded DNA, and that's normal for cells. Most of the time, our DNA is double-stranded, but sometimes our DNA is single-stranded, and some viruses, depending on the family, can have their genome single-stranded DNA. And our DNA in our host cells, our cells is single-stranded whenever the DNA is replicating or transcribing. The genome can also be RNA, and if it's RNA, it's double-stranded RNA. That is unheard of in cells. Or it can be single-stranded RNA, which that's normal in cells. If it is single-stranded RNA for the genome, it's important to state whether it is a positive single-strand RNA or a negative single-strand RNA. The difference is the positive single-strand RNA can be read by the ribosome, so it will have AUG coding the start codon, and then the ribosome will read this genomic viral RNA and use it to transcribe and make protein. Not transcribe, translate and make protein from it. This negative strand, single-strand RNA is the complement to the positive strand, so it does not have AUG. The ribosome cannot read negative single-strand RNA. The virus's genome can be either linear or circular. If you remember, humans and all eukaryotes have linear genomes, their chromosomes are linear, but bacteria, as well as mitochondria, have circular genomes. So that's, I don't know, normal. And then some viruses, like influenza virus, can have their genome in several segments. That should be familiar to you because humans have their genome in several segments, and we call these segments chromosomes in humans. In viruses, not everyone calls them chromosomes. Actually, few people do. So we'll just call them, the genome is in several pieces. We'll call them segments of a virus. Any question about any of that? All right. Viruses are surrounded by a protein coat. Let me blow this slide up. The protein coat shown here, with this side removed, can be, actually is composed of capsomeres. And there you can see each of these little circle is a capsomere. This virus, its protein coat is composed of one capsomere, meaning the capsomere just repeats many times. I have no idea. That looks like, I don't know, 20, 30, maybe even 50 for each side. And then there's, I don't know, 16, 20 different sides to this virus. And then this side isn't even shown. The capsomeres are proteins. They can be the same or several different proteins. In this case, it's the same protein that makes up the capsomere. And then the capsomere makes the entire protein capsid. So there's only one protein in this viral capsid, the protein coat. However, that doesn't have to be the case. Let me move on to show you another virus that has different. Here we go. Let's move on even further. This virus has at least one protein making the head portion of the virus, and at least two proteins making the sheath, probably another protein making the base plate, probably another making the pin, and then another protein making the tail fibers. So there's at least 1, 2, 3, 4, 5 different proteins making the protein coat of this virus. Let's see where we were about there. If the virus is only the protein coat and the viral genome, meaning there's no envelope, we call that a naked virus. So this would be a naked virus. There are some viruses, let me blow this up, which are enveloped, and here we're seeing an envelope virus. There you can see the genome right there, that strand, and there, right there, you can see the protein coat of the capsid. And then this right here on the outer portion is the envelope. Actually, the envelope is a bluish circular thing. The spikes are attached to the envelope, and they're the purple things coming out of the envelope. The envelope is a lipid layer, although it does have some proteins and carbohydrates in it. The envelope is external to the capsid of all viruses that have an envelope, but only some viruses have an envelope. The envelope may be covered with glycoprotein spikes. To my knowledge, all of them do, but that doesn't mean that all envelope viruses do have protein spikes. These are usually glycoprotein spikes protruding from the envelope. And because they are the outermost portion of the virus, these spikes have the antigens that the host immune system will recognize and then mount an immune response against. So the antigens are the outermost regions of the virus that the host immune system will recognize as being foreign and then mount an immune attack against. The antigens, or the spikes, also serve the virus as the binding point to the host cell. So once again, the outermost molecules of a virus are its attachment molecules for binding to the host cell. So in the case of the spikes, they serve as both the antigen and as the ligands or adhesions that attach to their host cell. Any question about any of that? All right, here's a question for you. On a naked virus, what molecules are serving as the adhesions for the virus to bind to the host cell? And they also will be serving as the antigens that the host immune system will recognize as foreign and then mount an immune attack against. I gave you a hint, and that is that will always be the outermost molecules. The outermost molecules will always be serving that role, which is a dual role, as both the antigen and as the adhesions of the virus. So what's the outermost molecule on a naked virus? The outermost molecule is the protein coat. And that will serve as the adhesions for this virus, as well as the antigens that the host immune system will respond to if the patient becomes infected with this virus. Any questions about any of that? All right, I stopped my share to see if someone was actually there. Let's go on in the show, though. Make sure I finish that. Yeah. All right. When we're talking about viral structure, viruses come in 3 different shapes that we call general morphology. And viruses are classified on their shapes. There can be helical viruses shown here. I don't see the helix there, but I'm just going to skip that. And I don't know why that's a helical virus. I'm sure it is. I just don't see the helix. There's a polyhedral virus, and that is many-sided viruses, such as this one here, the sectional electron microscopic image, and you can see the polyhedral side of the virus there. And that's the artist's picture. And then the third shape is a complex virus. And as you can notice, the complex viruses can take on essentially any different shape. Like this one here, doesn't look at anything like that or anything like that. And these are all complex viruses. Any question about any of that? All right. We're going to talk a little bit about bacteriophage T4, shown here, because it is the best understood, the most well-studied virus of all viruses. And the reason why it's the most studied and the best understood virus is simply because it is a virus which infects E. coli. And so early virologists decided to study it in detail to try and understand viruses. So bacteriophage T4 happens to infect E. coli, and they decided to study this virus simply for the reason that E. coli was a bacteria that was in many microbiology labs. Any question about any of that? All right. So bacteriophage T4 belongs to the family of T-even viruses, and they all have this shape. There's an actual electron microscopic image of a probably T4, but it could be a T-even virus. You'll note they have a head that contains the DNA inside of it and a tail fiber, which has different parts, the sheath, the base plate, and then the actual tail fiber itself. The tail fiber parts, this right here and this right here, is where the virus binds to their host cell. If the virus has a tail fiber, it's because the virus has that tail fiber to help it infect its host cell. And bacterial viruses that we call bacteriophages are viruses that infect bacteria. Because bacteria have a cell wall, many, if not all of them, have a tail to help the virus infect the cell. And the tail helps the virus get through the cell wall. Any question about any of that? All right, so let's talk a little bit about viral infection and the host immune response. Infection with a virus of an animal cell stimulates the animal or the host to produce antibodies. The antibodies recognize and bind to viral surface proteins that we call antigens, the molecule that the host cell or the host immune system recognizes and then mounts an immune response against. The antibodies will bind to those antigens and then attack the virus. Some viral proteins can change or mutate frequently, allowing different strains of the virus to occur, which can then infect the same individual more than once. A very good example of this is the influenza virus. And it has its molecules, especially the antigens, changing every few years, such that, let me show a picture of that. Once you get immunized to the virus, that immunization will only last about 3 years. So on the x-axis, we have time. And then the percent immunity on the y-axis, I'm not going to draw it because I can't, well, I guess I can just go like that. Percent. I'm going to abbreviate it. Percent immunity on the x-axis. So when you're vaccinated, you initially have a high immunity, but over time, that immunity drops. And once it drops below a certain threshold, they say you're no longer immune to that virus and that you need to get another shot. Well, that threshold for the flu virus is about 3 years. And that's as long as the vaccine will last. Beyond that point, they say that this is not, I shouldn't draw that in. I didn't mean to make the curve like that. So let's remove that part right there. But the point is, in reality, the immunity is linear, not linear, it will follow the same pattern. So you do have some immunity left. It's just below a certain threshold. We would say that you're no longer immune to it. And that's important to realize that even after 1 year, your immunity has dropped. And after 2 years, it's really dropped. Okay. But you may still have some immunity by 2 years. And like I said, I don't know what this number is here, but there is a threshold that they say that you need to get another shot. I typically get the flu shot every year because that maintains a high level of immunity. And you don't need to get it every year. You do need to get it at least every 3 years, because if you were to do it every 3 years, your immunity is so low that most of the time you would not have immunity to the flu after 3 years. And that is just showing you that some viruses can mutate frequently such that the immunity drops. With COVID-19, the virus does mutate fairly frequently. Not quite as rapidly as the flu, but it does mutate rapidly. And that's why they say you should get a COVID-19 shot about every year. And then a COVID-19 is, of course, more severe than the flu, although you're young people, so it's not so severe among you guys. But to maintain your immunity with COVID-19, you do need to get a shot every year because it too mutates fairly rapidly. Fortunately, most viruses do not mutate that rapidly. For an example, let's talk about the polio virus. Both of the polio viruses, not polio virus, both of the polio vaccines were made in the 1950s, sometime in the 1950s. I think one was 55 and one was 58, but don't quote me on that because I'm not sure exactly when, but sometime in the 1950s. And we're still using that vaccine today because the polio virus does not mutate frequently. And so the polio virus vaccines, which were made in the 1950s, and how many years ago is that? That's close to 80 years, 70 to 80 years ago. And the vaccines are still good. Any question about any of that? Sorry, I have to blow my nose here. I'll try and get out of the screen. Viruses are grouped into families based on the type of nucleic acid, their replication strategy, their morphology. You can group them into different higher orders. Let me see what's higher than a family. King Peter's son came over from. So viruses are grouped into orders, but there are few viruses grouped into anything higher than an order, except for we do group viruses into the kingdom that they infect, meaning we do have animal viruses and plant viruses. So we can group them into the kingdom that they infect. But other than that, we do not group them. Much above family, there is orders that you can be grouped into. And the reason why we don't group them into higher classifications is that viruses have a very small genome that evolves very quickly to match their host immune system. I mean, match the host cell. And so viruses evolve very quickly and have a very short genome. So we don't really know the evolutionary history of viruses much beyond their order. So we don't classify them between kingdom and order. We do classify them into family, genus, viral species, and subspecies. Grouped on, obviously, their genetic similarity, meaning we'll sequence the DNA. We also group them on their type of nucleic acid, the replication strategy, and their morphology. The virus family name ends in viridae. The genus name of the virus ends in virus, and the viral species is a group of viruses sharing the same genetic information and the same ecological niche, meaning they will, if they're in the same species, the virus will affect the same host and infect the same host species cells. You will be pleased to know that when we classify viruses, sorry, I'm losing my voice here. Viruses have common English speaking names. We have no Latin names for any virus. So all viruses, their naming is by their common English name. So the polio virus, that is its virus name. Any question about any of that? Let's talk a little bit more about the polio virus to talk about the subspecies. A subspecies, when we're talking about humans, would be the different races of humans. So there's the blacks and the whites and the Asians. Those would be the different races. And that's a different subspecies. They are members of the same species, but they have some differences. In viruses, the subspecies can be very important, at least as far as causing disease. For example, in the polio virus, subspecies 1 of the polio virus causes the polio disease. And you know what subspecies 2 causes? Anyone want to take a guess? Do you think it causes polio virus?
No idea.
No idea. The subspecies 2 is the oral polio vaccine that we give to prevent getting polio. So one subspecies is used as the vaccine to prevent getting the disease caused by the other subspecies. That tells you how important the subspecies are among viruses. The subspecies, when you give it a name like herpes simplex for the species, you just give it a Roman numeral for its number. So there would be subspecies 1 and then subspecies 2, or that would be herpes simplex type 1 and herpes simplex type 2. Any question about any of that? All right. So when we're talking about viruses, like the herpes viruses, an example would be the families. We call it the herpes viridae family. The genus is the herpes simplex virus. That's its common name. And that's the name I'm going to use because that name is continuing to be used, especially in the clinic and by Americans. They did come up with the scientific names for herpes simplex virus, and they said you should call it the herpes or the human herpes virus. However, that name has not stuck, even among doctors, and so I'm not going to use it. Human herpes virus, we will call it the simplex virus. And it has two subspecies, human herpes simplex type 1 or human herpes simplex type 2. Any question about that? Oh, excuse me. I gave you the old name. I mean, the new name. The old name, which is where you hear it, would be the simplex virus, or herpes simplex virus type 1 and the herpes simplex virus type 2. Any question about viral taxonomy, viral naming? Like I said, you don't have to learn any Latin names. You just learn the English speaking name. And surprisingly, apparently all of the early virologists were English speakers. And then later, when other people started studying viruses, they adopted the English names for the name. So the Germans and the French, apparently they weren't early studying viruses. And then when they started studying, they just adopt the names that the English speaking world had adopted. And that's good for us English speakers, by the way. When you're growing bacteriophages in the lab, the viruses must be grown in a living cell. A bacteriophage is a virus which infects bacteria cells. And you can pronounce that bacteriophages or bacteriophages. Both pronunciations are correct. And what you do is you grow them up in cells. So here, let me blow this slide up. I've actually done this, growing up bacteriophages on a plate of agar, and we coated the plate with bacteria, and that's why the agar is this off-white color right here where there's no viruses. And then you put on a solution that contains the bacteria viruses, the bacteriophages, and streak it around. And wherever there's a bacteriophage, it will stick to the bacteria and then replicate, lysing that cell and going out to neighboring cells, infecting them, replicating, lysing that cell, going on to neighboring cell, and that will give rise to the plaques, which we can see here. Oh, I don't know how many, but there's probably about 200 different viruses per sample, which was either 1 mil or 1/10 of a mil put on this plate. And there you can see that the virus probably immediately attached to a bacteria and began replicating. Why that's a very large plaque. And then there's a very small plaque. There's another very small plaque. What happened here was the virus went near a bacteria, but it had to wait until the bacteria replicated. and then replicated again, and maybe even again. And then eventually a bacteria grew to touching the plaque, and then the phage, and then the phage replicated in that cell, and then began the growth of the plaque. And that's why that plaque is smaller than that plaque or that plaque. This plaque is probably 3 plaques that fuse together to make 1 plaque. That's the best example I can see. Let me shrink that down, and then it's more apparent that that's 3 that grew together. Any question about any of that? So what you do is you put the bacteria, let's say it's E. coli, and you streak it on the plate everywhere so it grows in a lawn. And then before you incubate the plate, you put on a solution containing the viruses, the bacteriophage in this case, and then you streak it around the plate, and then you incubate the plate, and the bacteria will grow, and the viruses will grow, and as the virus grow, they'll form a plaque. The plaque is what the agar color is because there's no bacteria in that plaque, and so it's darker. Any question about any of that? And the reason why there's no bacteria in there is because the virus lysed all the bacteria in the plaque. All right, like I said, I've grown bacteriophages that way. Viral multiplication, how viruses replicate. The viral genome only contains a few genes, such as the capsid protein genes, maybe a few enzymes if the viral genome encodes and needs a few enzymes. And then the virus hijacks the host cell they infect. Following infection, the virus takes over the host cell's metabolic machinery and uses that machinery, including the host cell's ribosomes, the host cell's tRNA, the host cell amino acids, the host cell's ATP, some of the host cell's enzymes, to replicate the virus. The basic mechanism of viral multiplication is similar in all viruses. Bacteriophages, also called shorthand just phages or phages, are viruses that infect bacteria cells. So these are viruses that infect the kingdom, or in this case, the domain bacteria. Bacteriophages use 2 mechanisms to replicate. There's the lytic life cycle and the lysogenic life cycle. The lytic life cycle, the virus will infect the host cell, replicate, and then lyse the host cell, killing the host cell. In the lysogenic life cycle, the virus will infect the host cell, but the host cell and the virus will remain in symbiosis for as long as the virus stays in the lysogenic life cycle. That host cell will remain alive. Any questions about any of that? So bacteriophages follow 2 life cycles, the lytic life cycle and the lysogenic life cycle. To make new bacteriophages, what do the bacteriophages need in order to replicate? What components need to be made for a virus to replicate? I'm just going to show it here. You have to replicate the genome. And if we have double-stranded DNA, that's pretty easy to replicate. You just make more double-stranded DNA. You then transcribe that DNA to make viral messenger RNA, translate that viral messenger RNA to make the viral proteins, especially the capsid proteins. The capsid proteins then get together with the viral genome to make the new progeny virions. This is the minimum you need in order to replicate a virus. And obviously it will depend on what the viral genome is. We'll talk about that later. changing the viral genome. The virus, if it follows the lytic life cycle, occurs in 5 different stages. The 5 stages are: the virus has to attach to the host cells, the virus has to penetrate their host cell, there has to be viral biosynthesis, there has to be viral maturation, and then the viruses have to be released. Let's watch a little video on the viral lytic life cycle. Oops, that's not going to work.
First, the phage particle.
Let me see if I have a bad feeling about this. I don't think I can get this video to work in another player, because I don't think I have another player. I just don't think there's another player on this computer. I'll try Google Chrome. I don't think that'll work. Nope. All right, I can get it to play. Maybe what we'll do is we'll watch this one, and I'll just watch the beginning of this one.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately.
All right, let's stop that now.
Sometimes.
All right, any questions about the lytic life cycle? Let me back that up. And we'll keep that open because we'll get to that eventually. Let's see, where do I have that? There it is. So first, the virus attaches to the bacteriophage, attaches to the host cell. And once it attaches, the virus actually contracts, where the tail sheath, the virus penetrates the virus is that the tail sheath contracts. That puts pressure on the head, and that will inject the contents of the head into the host cell. And as the video explained, let's go back to that video. Where is that? Here it is. Come on. I might need to play it to show.
Attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria.
Remove this part of the cell
Next,
wall.
the phage breaks down the cell wall.
How it does that, oops, that's not what I wanted to do. How it does that is it,
In a way, a virus is a parasite.
how it does that,
It attacks a host.
how it does that is it actually secretes the enzyme lysozyme to break down the cell wall right there.
This attachment is very specific, and each type of certain type of bacteria, and the nucleic acid from this capsule.
And there you saw the sheath contract, that puts pressure on the head, which injects the nucleic acid into the host cell wall. That's what I was trying to show you. That works almost like a syringe, where when you push on the syringe, you inject the contents in the head of the syringe into the patient. And that's similar to the way when the virus tail sheath contracts. And then the virus will engage in biosynthesis, taking over the host cell machinery to produce its own genome, in this case DNA, and also will transcribe the DNA to make viral messenger RNA, to translate that viral messenger RNA to make proteins. How that often happens is the cell's machinery is used to do all of that. Although the virus could have an enzyme for replicating the DNA, meaning it brings in its own, what is that called? Not helicase, but drawing a blank here. Whatever enzyme copies the DNA, the
Polymerase?
polymerase, thank you. It may encode the polymerase. However, the at least the ribosomes and the tRNA, that's all supplied by the host cell. It could bring in the polymerase to replicate the DNA. It could even bring in the RNA polymerase to transcribe the viral messenger RNA, but it may use the host enzymes, the host polymerase, and then the host RNA polymerase to do that. It depends on the virus. Another thing the virus does, I don't think the video showed you that, is how the virus takes control of the cell is frequently what the virus does. That's not it. Where do I have it? There it is. It will degrade the host chromosome. And so that oftentimes
Is injected through the hollow
thing
tail into the cell.
virus does.
Enzymes and organelles.
Yeah, it's not showing you here. What it will do is it will, one of the first genes transcribed on the viral genome is it'll make an enzyme which will then degrade the host chromosome. The reason for doing that is the viral genome wants to run the show in the cell. And if it degrades the host chromosome, then only the viral genome is left to run the show in the cell. And that's just an easy way of gaining control of the cell. Any question about any of that? The viruses then assemble into new virions. What happens is the viral protein coat actually self-assembles, meaning when one protein comes up to another, they will start forming the viral protein coat until all of them are there to make the entire viral protein coat. And then there's an enzyme which stuffs the viral genome into the protein coat, and that's usually encoded by the virus, meaning the virus doesn't usually use a cellular enzyme to do that. It makes its own enzyme. And then the virus is fully mature. And then the last thing a virus will do, if it's following the lytic life cycle, is it will secrete the enzyme lysozyme, which will degrade the host cell wall, which will eventually cause the host cell to burst. And then that will release the newly replicated viruses, which we call virions. Any question about any of that? So there, the capsid attaches to the cell, and then it literally contracts like that, that forces the contents of the head into the cell. And when it contracts, the tail sticks itself a little bit through the cell membrane, which will allow the virus to inject its content inside the cell. Two terms you need to understand in the lytic life cycle are the burst time. That's the time from the initial attachment of the virus on the host cell till the time when the cell bursts from viral replication and the lysing of the cell. This averages 20 to 40 minutes, depending on the species and the temperature that you're growing the virus at. The burst size is the number of newly released virions. That's usually somewhat between 50 and 200 newly released virions. There's two terms you should know in the lytic life cycle. All right, any questions about the lytic life cycle? If not, let's begin the lysogenic life cycle. I'm not sure I'm going to finish this, but we'll try. Upon attachment of a host cell by a lysogenic virus, once the lysogenic virus injects its genome, which we'll say is DNA in this case, into the host cell, two things can happen. The virus can follow the lytic life cycle, so a lysogenic virus can enter the lytic life cycle and become a lytic virus, where it will go through the lytic life cycle that we just discussed. Or the virus can choose to enter the lysogenic life cycle. So a lysogenic virus has to make a choice immediately once it infects a cell. And that is, does it want to enter the lytic life cycle or does it want to enter the lysogenic life cycle? If it enters the lysogenic life cycle, the DNA portion of the virus will actually insert into the host cell DNA and begin the lysogenic life cycle. While the virus is in the lysogenic life cycle, the phage or the bacteriophage will remain inactive, meaning the latent or the lytic life cycle genes will be inactive. And why that happens is that the lysogenic life cycle has a repressor protein, and as long as that repressor protein is working, it will repress the lytic life cycle genes, which mainly are the proteins to make the new virions of the virus. The bacterial host cell is then termed a lysogenic cell, and it has its own host DNA, and inside that host DNA is the viral DNA. And it remains alive as long as the cell remains in the lysogenic life cycle. And we call this lysogeny. Okay? So when the cell enters the lysogenic life cycle, we call it lysogeny. And this will be the video I'm showing you now. The first part of the video gave you the lytic life cycle, but immediately upon entering the cell has to choose between, do I want to enter the lytic life cycle, reproduce, infect another cell. Once the cell bursts, it infects another cell, and then a choice has to be made. And if it enters the lysogenic life cycle, we then follow that life cycle. And let me show that video, which we started someplace. Oh, here it is.
To replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions about that video? Not, let's begin. Close that down. So what happens is the viral genome actually integrates into the host cell's genome. And so the host chromosome now contains part of the viral genome. That we call a prophage. So when the viral genome integrates in the host cell chromosome, we call that a prophage. Now, the only time the viral genome replicates is when the host cell chromosome replicates. And then that cell will divide. Each daughter cell will contain the viral genome in the host chromosome. Any questions about that? And that is the only way the virus replicates in the lysogenic life cycle. When the cell replicates, the virus replicates its genome. And that can go on for many cell divisions where that occurs. So one cell can become 2, 2, 4, 4, 8, and that can continue. So there's 100 or thousands or millions of cells. And then for reasons that are not fully understood, the viral genome will come out of the host cell chromosome and then enter the lytic life cycle. Always when the virus switches from the lysogenic life cycle to entering the lytic life cycle, the viral genome will excise itself from the host chromosome. The reasons for the virus removing itself from the host chromosome and entering the lytic life cycle are not well understood even today. And they've been studying this for over 100 years, and they don't know why most of the time the virus leaves the lysogenic life cycle and enters the lytic life cycle. They have learned one reason it may leave the lysogenic life cycle and enter the lytic life cycle, and that is when this DNA gets damaged, the virus will somehow know that the host chromosome is damaged and that the cell may die, in which case the virus will leave the lysogenic life cycle and enter the lytic life cycle. And if it enters the lytic life cycle, that cell will then reproduce new viruses and be lysed, and the cell will die. Any questions about any of that? All right, not hearing any questions. I think this is a good place to stop. And it's true, we're behind schedule, but let me briefly finish the lysogenic life cycle. I already talked about that. I already talked about the repressor protein keeping the cell in the lysogenic life cycle. I need to talk about the consequences of lysogeny. There are two consequences if a bacteria cell enters lysogeny, meaning it has a lysogenic virus in its chromosome. First, the cell will be immune to infection by other species of that species of virus causing the lysogeny, meaning no other species of that virus can infect the cell. So once the cell has become lysogenic, that same bacteriophage cannot infect the cell further. The other thing that can happen is this cell picks up new genes. The genes are the genes of the virus, and that can change the phenotype of this cell, and that we call phage conversion. When the cell changes its phenotype by the genes of the virus. One example of that is Corynebacterium diphtherium only produces the Corynebacteria diphtherium toxin when it is a lysogenic cell, meaning it only produces that toxin when the Corynebacterium diphtherium has a lysogenic virus inside it. Any question about any of that? So yes, depending on how sick you will become may depend on whether the bacteria that has infected you is infected with a virus. Any question about any of that? Actually, that's really good. I finished lysogeny, now we're talking about animal viruses, and when we come back, we'll talk about animal viruses. And that won't be until next week. I will be logging in briefly for 15 minutes to answer questions in the lab, but other than that, there's no lab today. All right, there's no questions, I'm ready to go.
Yeah, I just, when can I expect this video to be posted on your YouTube site? I just use the videos to help me decipher my notes.
I'll try and do that sometime after the lab today.
Okay, perfect. Thank
Okay.
you.
All right,
Bye.
bye.